at Wildlife Forensic and Conservation Genetics Cell. Dr. Gupta is a master's in biotechnology and did his PhD in wildlife forensic from Saurashtra University, Rajkot, Gujarat. He is a JRF in a DPT funded project at the CCMB Hyderabad. Dr. Gupta has been conducting research in the area of conservation and forensics since 2002. His research interests are evolutionary and conservation genetics. His team is working on basic and applied research project for the conservation of rare and endangered species. He is also undertaking customized teaching and training courses on the illegal wildlife trade. Dr. Gupta is currently working on many projects, and some of them are development of molecular markers for wildlife conservation, applied conservation genetics of tiger for population management and enforcement, evolutionary and phylogenetics of endangered animal species. Instead of this, he has examined more than 2,200 wildlife crime cases in his career. Along with this, he has contributed the various book chapters within the different publications. Dr. Gupta is a guide and co-guide in many research work, and currently 13 students are enrolled under his supervision. So with this short introduction, I would like to call Dr. Gupta for the deliberation. Over to you, sir.
Thank you. Can I share my screen? No.
Yes, sir. Please.
I cannot share my screen.
Yes, sir. Please share the screen.
No, it is not able to. It is disabled.
Technical team, I request you to enable the sir to share the screen.
आलोक सर, सर को होस्ट कर दीजिए. संदीप गुप्ता सर को होस्ट बना दीजिए. को-होस्ट.
यस सर, क्या करना है?
संदीप गुप्ता सर को को-होस्ट कर दीजिए सर.
संदीप सर को ना?
हाँ जी सर. Sir, now you can share your screen.
Is it visible to everyone?
Yes, sir. Yes, sir. Visibilities.
Right. So as it was introduced that I am working in this field of wildlife forensics for more than 20 years. So I started my career from CCMB Hyderabad and then joined WIS faculty and now heading the wildlife forensics division of the institute. When I understand that this program is for the forensic science, but when we discuss about the wildlife forensics, it is very specialized branch of the wildlife forensic science. Though the forensic science is a century-old technique people are using in the field for the legal purpose, but wildlife forensic is introduced at the end. So it is very new branch of the forensic science. It is not a more than 3 decade old practice per se. If you have little bit background about the conservation law, so in our country it was implemented in 1972 with the Wildlife Protection Act. Before that, there was no legal issues in hunting these species, not very severe issue. But after 22, under Wildlife Protection Act, there are punishment decided for the hunting, or not only hunting, but possession of the wildlife article also become illegal here. So whenever anyone, any culprit, any accused is found with any article which is suspected to be of wildlife origin, so as per our country's law, it is punishable offense. There are many countries, those do not say it is offense, but they do regulate the harvesting of the wildlife from the field. So which include the several African countries, US, and many other part of the world where hunting is a licensed practice. People get the hunting license, they go in the field and hunt the animal and then consume its part based on their licensing terms and conditions. But in our country, it is totally banned. There is no any case hunting is allowed for the joy purpose. People are not allowed to hunt any species on routine, on regular basis, you can say. But removal of animal is allowed in certain condition, and that too is permitted by the chief wildlife warden of the state. So those are the very specific conditions when animal is allowed to be removed from the field. Removal doesn't say that it has to be hunted. Essentially, it can be captured and relocated to a safe place where it should not harm like it was harming earlier. So there are many crop raiding animals. Those are declared vermin time to time by the state forest department through chief wildlife warden's order. And then sometime if animal become threat to human life, so then also there is a provision to issue the permit. Otherwise, except these two situations, there is no regular practice in our country for removal or hunting a species from the field. So that is why, so here, any wildlife trophy, any wildlife article is illegal if it is not registered in Chief Wildlife Warden Office. So when we talk about the wildlife conservation, in global perspective, our country is a leader in conservation. Though having very small land share in the globe and very heavy population, human population, so we are going to become a #1 populated country in the world very soon. But still, we are able to manage wildlife in wilderness condition in our country. So it is a problem for the countries where biodiversity value is very high, but not only limited to such country where wildlife health is very good, but it is also a problem for the globe. Entire world is suffering through the wildlife crime or illegal wildlife trade, especially when it comes in organized trade. So it comes at 4th number after illegal arm, narcotics is first, then arm, then human trafficking, and 4th one is the illegal wildlife trade. And only wildlife trade is earning a revenue of about 20 to 40 billion US dollar per annum, which is very huge value. And not only pertaining to the biodiversity loss due to hunting, but also in various situations as money laundering object. And we cannot forget about the recent pandemic situation which we are facing, the COVID, which has direct linkage with the wildlife origin. So there are natural COVID host in the wild condition, and when any situation arises where natural host release the pathogen for the human disease, it become a emerging disease like COVID pandemic. So everyone knows about now. We don't need to educate people for the zoonotic disease. Everybody knows that how worst it can happen. So that is also another thing which can easily transport, easily easily transported through the wildlife article from one place to another place in no time due to recent travel mode and spreading mode. So within a day, people can reach from one corner of the globe to another corner. So that is the speed of even transmitting a disease from one corner of globe to the another corner. Earlier, when travel mode was not such developed, then spreading disease was very poor, very low, and it was moving through the human carry. But now it is spreading very fast, like it happens in COVID situation. And when we talk about the wildlife offense and identification of parts and product, so it is very, very difficult for the even expert to identify each and every object. So expertise is not up to the mark. Everybody cannot say that he is perfect expert of wildlife article. So it is still growing phase and people are still learning from the example. There are variety of objects and variety of derivatives all across the globe. Those are in demand, especially wildlife meat, bush meat in many countries, especially in Southeast Asia. Many wild species are being eaten in street as street food. So you can observe them. People are eating snake, lizard, frog, cockroach, scorpion, every species they do consume for their taste. And then there are many articles which are being used as a desiccated and dried condition for the medicinal purpose as well as making the ornamental trophies. If we talk about the conservation status of our country, I will give one example of tiger. So global tiger population is estimated about 4,000 in entire tiger-rich country. And of these 4,000, about 3,000 tiger occurs only in our country, and remaining 1,000 tiger are distributed in other countries, including Bangladesh and Siberia and Russia. So you can imagine that how well we are doing for the tiger conservation. And then there are captive bred tiger also in China, because China are heavily dependent upon tiger body part for their medicinal purpose, especially tiger bone. So there are variety of species, those are in illegal trade. And top priorities for the cat, feline, then elephant, deer, rhino, bear, musk deer, and coral insects, and then pangolin is again another species. Whenever any article is linked with their medicinal property, its value goes many-fold high. So that's how happening with the wildlife article, especially in China. So they use each and every wildlife article for the medicinal purpose. And that is why its cost is very, very high in the Chinese market. Then there are many agencies, those are working for the law enforcement, including Wildlife Crime Control Bureau, State Forest Department, Police, Custom, paramilitaries. And all these agencies are primarily looking for the implementation of the Wildlife Protection Act. Then many other acts and CITES together can be implemented. In our institute, we started wildlife forensics in 1995 with the support from US Fish and Wildlife Service. It is still global leader in wildlife forensics. Its laboratories in Ashland in United States. So in 95 we started with the project mode with USFWS. Then 1998 we established our lab, and 2017 we were recognized through Gazette notification. So now our report is taken under CRPC section 293 as a admissible evidence. This is annual record of the receiving cases in our institute. So about 300 cases we do receive on every year from entire country. So you can send the country's map that samples are sent from all across the country. It is not only from the one region. And there are many ways to deal with to identify the wildlife particle. It could be a very conventional morphological based protocol and the advanced one, the molecular testing. So we do utilize both the technology here, morphology as well as DNA. So DNA based cases can be built up to 65% and remaining 35 to 36% cases are dealt using morphological tool. They don't need any molecular test. So DNA is a very simple approach for identifying a species from the unknown article. So for that, we first have to establish a good repository. Then we have to work out on the protocol for the extraction of the DNA from variety of the wildlife articles and marker. We need to identify which marker is suitable with minimal effort. Minimal effort is very essential because there are more than 1,500 species and we cannot go through a species-specific approach. We include a universal marker which deal all the species all together. So we don't need to go with one by one species testing. Sample can be preserved. Sample can be preserved in ethyl alcohol, especially in 70% ethanol. Formalin should never be used. बताओ अच्छा बैठो. Formalin need not to be used as preservative. And for short-term storage, we can even use silica gel. And blood in the various form of the stain. So we can use blood stain from the stone, leaf, litter, log, and many other objects. So it can be used as it is for the species confirmation. Approach which we use, we extract DNA from the sample, then amplify using universal primer designed for the mitochondrial DNA. And then we do the sequencing of amplified PCR product. And then we compare the sequence in our database. And based on comparison, we conclude our result. It has helped in identifying various objects like this Hatha Jodi was sold as plant root on various online shopping sites. When we procured and checked, it turned out to be a biological sample of Varnus bengalensis, which is monitor lizard. So it was basically a male genital organ of monitor lizard, which was sold as plant root on every shopping site. Even monitor lizard's complete skin used for making handbag. Then a gland of American beaver, which is found in North American Canada, has reached to our country. Then antler of fellow deer. You can see the last four antler are very unique. And those were of the fellow deer. Those are found in Europe. So, and it was recovered from Maharashtra. So we are getting a lot of exotic species even from our country. It was a very famous case where one of the sport person was involved, former sport person who was captain also our national team. So he was alleged to hunt a black buck, and in his case, blood stain trial, blood stain knife, and blood spot from his vehicle were sent to me for the confirmation. And he was alleged to hunt black buck, and when we checked, all the blood samples were molecularly identifiable as a black buck, and we reported that. Even it has helped in identifying individual of the tiger through confiscated claw. So from a claw, we could nail down that this claw belonged to a tiger which was killed 5 years back in a zoo park using mitochondrial DNA hypervariable region as well as DNA fingerprinting marker, which we called micropixelized marker. So using this different marker, we could identify that the claw were of the same family which tiger was missing from the zoo. Wooden chopping block, like in this case, has helped in identifying the bird which was chopped on that surface. So this chopping block was very clear and clean. And when we scraped the surface of the wooden chopping block, it gave the identity of peafowl. And remaining two objects which were recovered, like non-edible part of the bird as well as cooked meat, those two were matching with the chicken. But wooden chopping surface gave the identity of peafowl. I was pioneer in establishing a technique protocol for extracting DNA from the ivory, and I could identify two different ivory idols as a Asian elephant origin. The DNA-based test has helped in identifying the visceral content of the 3 leopards died in the same day, and their visceral content had the single dog remnant. It means all the 3 leopards died due to consuming a single dog, and that too matched with the dog carcass found around that area. So there was one half-eaten dog carcass also recovered. So we could make it out that same dog was consumed by 3 leopards, and after consuming leopard, they were died. And that dog was poisoned. Later on, it was detected that it was poisoned, and due to dog carcass poisoning, 3 leopards were died. Yeah. Pangolin is another species which is killed by boiling in water, and after boiling, its scale is separated out. And once the scale is separated, then its other body part is not thrown but consumed, and scale is used for trade. So there are huge consignment coming from India as well as from the abroad, especially in 3 ton, 7 ton, 12 ton, and 26 ton in quantity. And one pangolin entire body can give maximum 300 to 500 gram of the scale. So if maximum 500 gram is coming from one pangolin, then 26,000 kg is definitely coming from more than double the number, more than 52,000 animal, like that. Euromestis is another species which is boiled live in live flow in a cooking oil, and after it is disintegrated into the oil, so that oil is collected and sold as a sand oil. And morphometry is also another potent method of identifying a species, especially it can identify to differentiate fake versus real skin. So first 4 skin which you can see in this picture is fake, and the third skin only is the original, and in bottom also leopard case. So second and third one is the original, and first one is duplicate. So there are way to identify it. We do the hair microscopy for the mammalian hair, and under microscope we can identify each species. And there are claw also identified based on morphological feature. If we get the skeleton of the species, we can identify through morphological test, and even if we get the large bone, we can identify. So these are the conventional method, but here for the knowledge of the audience, I am sharing it. Another product is Shahtush, which is coming from Tibetan antelope, and it is a product which is called Shahtush. All is highly demanded in global market. Reptile skin is used for making necktie. Mongoose hair is used for making paint brushes. Musk deer, male musk deer pod is used as musk pod, and only male musk deer do contain one pair of very sharp canine. Bear is killed for its gallbladder, which is called bear bile. We have received the bear bile of three species. One is black bear, then cloth bear, and another is brown bear. So we have another species of bear in our country, which is found in northeast. It's called Malayan sun bear. So we have not got any bear bile so far from the sun bear, but still there is possibility that it is also killed for its gallbladder. Elephant was assumed to be hunted for only its ivory, but not limited to. There are many other artifacts and many other body part which is being used, like elephant tail hair, elephant leg, and in the palm of this lady you can see it is elephant skin bead. The bead made up of elephant skin is also used in jewelry. Antler is used for making various objects. Then canine, variety of species canine is used, and especially when crown portion of a canine is having groove, like in this picture you can see the groove, two line, and in this picture also you can see one groove. So if this groove is there, so it belongs to the felid. All cat canine do contain the groove line. So that is the identifying feature from felid from non-felid. Tiger, leopard will have this. Your domestic cat also will have this groove, but not any other species. Then a marine turtle, horsebill sea turtle, is also hunted for its black and yellow spotted carapace for making jewelry and ornament article. Rhino horn is harvested at mass scale, which is a reason for declining rhino population in the globe. So we have 3 Asian rhino and 2 African, black and white rhino in Africa, and Indian rhino, Sumatran rhino, Javan rhino in Asia. कहाँ जा रहा है? क्या बेटा? कितने बजे तक हमारा? Then fake rhino horn is also floating in the market. So there are many doubt arises when article become fake and original. So we need to differentiate them also on a routine basis. So you can see a lot of nail inside the keratinous, inside the melamine sheath. So this you can see visible here. Even elephant bone is also carved like rhino horn. A rhino horn is basically a bunch of hair. Our targeted effort is going to sensitize the enforcement agencies, especially we do give the training, hands-on training to the custom officers, State Forest Department, and the officers of Wildlife Crime Control Bureau Those are the enforcement agencies, but we also give the training to the scientific agencies like forensic scientists from the Nepal, Bangladesh, Bhutan, and now we have started giving training to the scientists from State Forensic Science Laboratory. So we have given a training to the Karnataka State Forest State uh, FSL forensic State Forensic Laboratories. So they now we need more and more working hand to help us. We can't cater the need for entire country, but we need many other working labs in the regional level so that the disposal of case can happen at very rapid rate. We even gone to the Interpol headquarters to train the European participants, high-level participants in identifying cytosol. We have established a protocol, laboratories, manual, and altogether a fully functional laboratory. Constraint will always be there. Expectations will be very high. But with all those constraints and limited support, we are able to support entire country for the wildlife forensic investigation. So this is brief about my today's talk, and we'll have more on discussion mode. And I expect there need to be many more questions from your end so that we can answer your query rather than giving my own input. So it will be better if you have any doubt, anything, you can clarify here.
Which category of poison are used to kill the animal? Okay, thank you so much, sir. Participants are requested to put the questions into the chat box.
Or participant may raise the hand also. They can directly interact with Gupta sir.
One question we have received from the Miss Hiramani. She is asking which category of the poison are used to kill the wild animals in India. I think she is asking about the trained, what kind of the category of the poisons are mostly used to kill the animals.
Generally, people do use the readily available poison. They don't look for any specific poison. Especially, they go to the pesticide shop and they procure the pesticide and they bring it and poison the carcass. So that is the major trend here.
Right.
And that even a laboratory which is functioning for the toxicological analysis can answer better. Our lab is basically for the species confirmation, so we do not cater at this moment for the toxicological study.
Another question we have from the Dr. Sangeeta Patle. She's asking,
No. Protein profiling is not a polymorphic, and we cannot identify species using protein profiling. That is very least polymorphic marker. So we need more and more polymorphic markers so that differentiation is easily done. So we need only molecular marker here.
So we can use polymorphic enzyme as a marker.
Polymorphic enzyme will also be not like very accurate. It may lead to you up to a certain group, but not up to the.
We cannot individualize the
not even species, not even species.
Okay. Anjali, madam, has raised the hand. Anjali. Alok sir.
Yes, ma'am.
Yes.
अंजलि मैडम को अनम्यूट कीजिए.
कौन सी मैडम?
अंजलि भार्गव.
इधर इसमें एक मिनट अटेंडेंस मांगो शायद. अच्छा जी. उनको ओके करने का बोलना पड़ेगा मैडम. इधर से मैसेज तो गया है.
ओके.
Okay.
Anjali, you may write the question in chat box also.
मैं उनकी स्क्रीन पर ओके के लिए मैसेज आ रहा होगा एक्सेप्ट करने के लिए. वो करना.
Yes, yes, but okay, no issue. Any other question from participants?
Okay, thank you so much, participants. Now I call to the Dr. Swati Dubey Mishra, madam.
Uh, Thank you so much, sir. Thank you so much, Dr. Sandeep Gupta, sir, for accepting our invitation and deliver lecture is on such an emerging field. Now, like as human forensic, advancement in the field of wildlife forensics is also very important to prevent and investigate the wildlife crime. We can take an example of Salman Khan black buck hunting case. In that case, DNA profiling has played a very important role. So apart from human forensics, we can use traditional as well as hyponetic technique to solve the wildlife crime. Thank you so much, sir, for your valuable time. Thank you once again, and we'll meet in another session. Thank you. Thank you.
Thank you so much, sir. With the due permission of the coordinator, convener, and the speaker, I announce us to close the session. Thank you.
Ayush sir. Hello, Ayush sir.
चलेगा.